I do have a slight concession this Saturday. Saturday, whatever, didn't know the date. So please bear with me today. And as you know, people who get concussions tend to be short-fused. So I am very short-fused. I've been yelling at everybody this whole weekend, so I'm trying to be nice. Like the whole drive, I'm like, be nice to your students, be nice to your students, all right? So bear with me, and if I do pass out, there's the phone right there. You dial 4 4s, okay, and that connects you directly to the campus security, and just tell them the room, and they'll take care of it. But I won't pass out, all right? And as I see, probably some people thinking it's a holiday. They decided not to show up today, right? So they're going to be screwed.
Extra credit.
It's not a holiday. All right. Last week we talked about Mendelian genetics. We did the analysis. We talked about segregation, assortment. And if I'm not wrong, correct me, we talked about incomplete dominance, right? Red and white in case you get pink, right? And then we talked about codominance in the case of the bloods and everything else, right? And then after codominance, we talked about the effects of genes. We talked about that one, right? Where a set of one alleles affects the influence another set. If it stops it, we call it epistasis. Or when a gene gives you multiple effects, we call that pleiotropy. And now we're going to talk about environmental interactions, right? With phenotypes. We're going to talk about interactions between environment and phenotype. After all, it's not all about genes. Genes are important, but the environment does play an important role when it comes to phenotype. The example I'm going to use is the Himalayan rabbit. Anyone ever been to Himalayan? Anyone ever seen a Himalayan rabbit? Mm-hmm. If you look at a Himalayan rabbit, the fur tends to be white, except for extremities. Tail, ears, the feet, the nose. Demonic Himalayan rabbit. Alright? So the fur, generally speaking, tends to be bright white. However, when they are exposed to cold, the cold will activate an enzyme. In other words, the cold will cause the DNA to make an enzyme that allow for formation of dark pigmentation. In this case, it's melanin in the hair. That's why, the feet, the ears, the nose, and the tail are black. It's getting cold right now, right? Mm-hmm. Which part of your body feels it the most? Your feet, your fingers, your ears, right? Extremities. So do you see the evolutionary advantage of this? Because the extremities are exposed to the cold air, causes the expression of an enzyme which allows for the dark pigmentation. But the rest of the body tends to remain warm. They did a study. They took this rabbit, shaved the back, and then they waxed it, right, completely, no hair. And then they put an ice pack on it. So imagine this is an ice pack. And they put an ice pack on it for a couple hours, and then they removed it and let the hair grow. Guess what color was the hair? It was black. The hair was black because the cold air triggered the formation of black fur. All right? So this is an example of interaction between environment and phenotype. Right? There's another topic we don't cover in this class. It's called epigenetics or epigenes. And pretty much the gist of it is how environment influences your gene. And plus, whatever you put in your mouth right now will influence your grandchildren. So what might be happening to you right now could be the result of what your grandparents ate or they were exposed to. Maybe they were younger, or their parents were exposed to. All right? So it does complicate the matter. All right. Any questions? So we are completely done now with classical genetics. Now we're going to move on to chromosomes, focus specifically on humans. We're going to focus on you guys. We're going to talk about disorders, which you guys can relate to. On the exam, this is important, on the exam, make sure you know all these disorders, what do they cause, what are the symptoms, and so forth, okay? But we're going to talk about human genetics and so forth. We're going to review some terminologies again, just to help you guys out. All right, so, but before we do that, let's start out with a puzzle. I'm going to use my family as an example. All right, so look at this. Hey, censorship. Look at this couple right here. Me and my wife. See how happy we were? This is before baby. This is actually we were happy because we're flying to Amsterdam. You can't be happier than that, right? You're happy flying there, and you're very happy flying out too. But you know, I have brown eyes, and she has hazel eyes, right? Now I want you to calculate the probability, and using Punnett square. Go ahead and do it. Of us having a baby with blue eyes. Calculate the probability using the, fraction and percentile of us having a baby with blue eyes. First of all, go ahead.
But are you like the heterozygous?
Well, you just figure that out. I'll leave that as a blank on purpose, okay? Just do it solo. Should we prank call the room? Hello?
Bigger than women, the two small A's.
And then one small B. And then the sound one to go.
Solo. I'm going to give you guys 30 more seconds. You have to nail these on the test. 30 seconds or less. All right, so look at me here. Now, first of all, is it possible for us to have a baby with blue eyes? Yes or no? Just say that.
No.
Yes. It is possible if we are both
Heterozygous.
heterozygous. In other words, we are both this way, right?
Mm-hmm.
One dominant allele and one
Recessive.
because as long as you have one dominant, you get the dominant trait. So if you were to do the Punnett square, assuming, right? These two combine. These two combine. These two combine. What eye color do I have here, or the baby will have?
Brown.
What about here?
Brown.
What about here?
Brown.
What about here?
Blue.
So there's 1 in 4, or 25% chance, right? This is only one square, right? So assuming that we are heterozygous, we have 1 in 4, or 25% chance having a baby with blue eyes. Now, if I know for a fact that I'm homozygous dominant, if I knew for a fact that me, homozygous dominant, is there any way for us to have a blue-eyed baby?
No.
Never. Okay, you see how that works? So keep these scenarios in mind. Keep these scenarios in mind. Now, of course, we did not have a baby with blue eyes. I can live with the disappointment. But this one, he was at 1 month old. I told him get a job, and that was his reaction. But notice, his eyes are brown. And those who said, don't let this demonic look fool you. He destroyed my wife. This is a woman who beat me in every marathon we competed. She can climb any mountain. Pretty much an elite athlete. She can walk faster than most people who can run. Seriously, that's why people were telling her she should be a runner. I come home one day, that's this last semester. This is what I see. She is passed out. And of course, he's in there thinking, going, swinging back and forth. But she was gone. I walk in the house, and I have to take a picture. But he was out, because he was a very difficult baby. Never slept, hour and a half every day, that's it.
Wow.
Hour and a half, woke up for an hour. Hour and a half, woke up for an hour. Like, demon child. I seen him hover once in a while. Let's hear it. And just wait for his baptism. I'm willing to bet the water will evaporate. Then he's going to start speaking Latin. All right. Let's do another problem. In pea plant, spherical seeds are dominant seeds and general cross between plants are heterozygous. So you get to see that, right? So I have a spherical plant crossed with a spherical plant. Calculate the percentage of the offspring that will be spherical and calculate the percentage that won't be spherical. So just go ahead and calculate that. Spherical and non-spherical. Good eye, sweetie girl. Should be able to do this in 15 seconds or less. So, calculate the percentage. What percentage will be spherical? Not enough to do the Punnett square. I want to practice so you guys can learn the interpretation. It's not that hard. Get my markers. I should write on this on purpose. Again, you do the Punnett square, right? You put one here, and the other one here. Oh, smaller. So you get homozygous dominant, heterozygous, heterozygous, and homozygous. Is this spherical? Spherical?
Yes.
Spherical?
Yes.
Yes. So 3 out of 4, or 75%, right? And then the one that is non-spherical is 1 and 4, or 10%. So you will have problems like these to solve in the exam, okay? You will have problems like these to solve. And I think most of you are doing well. You're doing well. And the Wednesday before Thanksgiving, you will have a quiz, all right? And it's going to pretty much deal with Mendelian genetics and some of this stuff here, okay? All right, so let's talk about chromosomes and human genetics. After all, chromosomes are extremely important. Now, this stuff, don't worry about it. I already give it. It's in the PowerPoint. It's posted, right? But what is exactly chromosomes? Pay attention. Chromosomes are rod-shaped filamentous bodies. It's pretty much, that's where the genetic information is located. And we saw those when we looked at the mitotic stages under the microscope in the lab, right? And I showed you pictures, right? They carry genes or units of heredity. And of course, they are not very visible in the active stage. They only become visible when they are inactive. And of course, you have to use dyes to see them, all right? I'm going to spend too much time on them. So pretty much this is what a chromosome looks like. You have the genetic information, DNA, coils around the protein. And then these proteins coil around themselves, and you coil, and you coil, and you coil, and you coil, and end up getting the chromosome, all right? So this small region is supercoiled. This is called supercoiling. You can take something that is 2 nanometers and coil it into something that is much smaller. Example I told you guys was taking, say, length of this. Imagine from here to there, to be able to coil it and put it into something that is this big. So imagine you have a rope all the way, coil it and put it in something this big. If you completely uncoil all the DNA, it not only will rupture the nucleus, it's going to fill out inside of the cell. Okay? So it's much larger, all right, supercoiling. There's some computer generation. These are actual pictures of it. I'm not going to spend too much time on it. And we talked about the duplicated chromosome. Each one is called a sister chromatids, right? Each one is called a sister chromatids. They were discovered by Strasburger, 1875. So go way back in time, all right? But they don't use the term chromosome until this guy uses it. Don't worry about his name, FYI, just in case if you get on Jeopardy one day, who knows? They might ask you these questions, all right? And chroma means color, soma means body. That's what the name comes from, chromosomes, because they have a high affinity to pick up the dye. So they're really easy to dye, to stain. And that's why they said, oh, they're stainable, so let's call them chromosomes. Right? But they're nothing more than DNA. I already talked about this. But if I ask you this question on the exam, why chromosomes are important to an organism? Why do you think it's important? Again, what do the chromosomes carry? Genes. And genes are nothing more than units of information about a specific trait. So again, why are chromosomes important? Go ahead. It's not a trick question, people. Say again? Yeah, but why is it important? Why are chromosomes important?
Because they carry information about different traits.
Every trait we have is carried by the chromosome. Right? Every trait is carried. Organism must have the exact number too, by the way. Later on, we'll talk about abnormalities. If you're missing a chromosome, Turner syndrome. If you have an extra chromosome, Klinefelter syndrome. All right? And of course, the normal chromosomes give you the normal conditions. We'll talk about this. But it's extremely important for you to have the proper number of chromosomes. If you're missing a chromosome, you might be missing something extremely crucial. All right? Now, how many chromosomes do humans have? 23 pairs. All right? 23 pairs. And of course, the number of genes vary between different species. Humans, we have about 20 to 25,000 genes. But we're not that special. There's some plants that have 20,000, 19,000, and so forth. So the number of genes vary, but again, the number of chromosomes are extremely important. Yes?
How do they know how many?
We sequenced the human genome. So they counted them. And we'll talk about that technique at the end of the semester.
Like they counted 1 to 20,000?
Yeah. There's a machine that actually does it for you to look at activities and stuff like that. So notice in this individual right here, there's an extra chromosome on number 21, right? This is called a trisomy 21, also known as Down syndrome. Okay? To have Down syndrome, what happens is you end up inheriting an extra chromosome on number 21. That's what causes Down syndrome. Okay? Extra chromosome. Now, how does this end up here? We'll talk about that later. Okay? We'll talk about that later. By the way, when it comes to the number of chromosomes, I mean, penicillium has from 1 to 4 depending species. Mosquitoes have 6, houseflies have 2, garden peas have 14 chromosomes, humans have 46, orangutans, chimps have 48. But look at this, adder tongue ferns. How many chromosomes do they have? Depending on species. That is a huge number. That is a humongous number. That's why plants can tolerate self-breeding. Because they have so many chromosomes, even if you're self-fertilizing, you're still mixing quite a bit of genome. Humans, technically, you're not even supposed to marry your own cousin. I mean, there's a reason we said don't marry your sister, right? Don't marry your sister, because genetically, chances of passing back traits gets closer and closer, all right? Tell that to West Virginians. Okay? But, theoretically speaking, even your first cousins you're not supposed to marry. Even your first cousins you're not supposed to marry. Because the closer you are to each other genetically, the chances of 2 bad traits combining is much higher. The closer you are to each other genetically, the chances of 2 bad traits combining is higher. That's why, if it's up to me, I think marriage should be a lottery system. Pick people from different countries, just give them numbers. All right, you two marry. It won't work that way, but you know. Right? And of course, humans and apes, you can't cross-fertilize because they have extra chromosomes. Now, Stalin had a scientist who tried to cross humans and gorillas. He wanted to create, I kid you not, this is Planet of the Apes stuff. He wanted to create a gorilla army. And they had females lined up for this process. Luckily for us, Stalin stopped trusting him and he ended up dying in a prison somewhere. Some mad scientist. But it would have never worked. It would have never worked. They have extra chromosomes. And when we talk about human evolution, I'll show you why there's a difference between them.
When you say that would have never worked, what do you mean, that she
It
wouldn't
would
have got
not produce, it would
pregnant,
not-
or wouldn't have had it, or?
I mean, you can force the sperm into the egg, but the mixing of the chromosomes, that cell would have not been viable. It would have not been viable. It would have just been destroyed. All right? Now, one of the first chromosomes that was discovered to have an association with a disorder was to have an association with cancer. All right? And the name of this chromosome is called the Philadelphia chromosome. A lot of times with the genetic disorders, they either name it after the place it was first described, or they name it after the person who found it, or the person who has the disease. Which I don't think is the right thing to do. It's like, oh, sorry, you have an Isani? You have an Isani, I'm so sorry. You know, I don't think it's right to name something after a person, especially when it comes to a disease. Name something nice. But Philadelphia chromosome was discovered in the Philadelphia region, and it's the first abnormal chromosome to be associated with cancer. In this case, it's chronic leukemia. And chronic leukemia is overproduction of the, remember leukocytes, white blood cells? Let's think of it as a blood cancer, all right? This is an example of a blood cancer. Now, what happens in Philadelphia chromosome is your chromosome number 9 and chromosome number 2 exchange pieces. Now, do chromosomes exchange pieces under normal conditions? Yes. Remember we talked about crossover? Remember that prophase? Homologous chromosomes line up and they exchange pieces, right? Does that ring a bell?
Mm-hmm.
But since they're homologous, that's not a bad thing. That's actually a good thing. But 9 and 22 are not homologous. So you're exchanging two foreign pieces together, and this will lead to an abnormal gene that causes the overproduction of the white blood cells. So Philadelphia chromosome was one of the first chromosomes to be associated with a disease. In this case, it was called cancer. Because before this stuff came out, a lot of the genetic abnormalities, you know, people are stupid. When I say humans are stupid, believe me, all right? Baby comes out abnormal. Baby has Down syndrome, all right? Oh, it's the mom's fault. She did not pray hard enough. Or she lied. Or whatever, all right? So when we were given these things, of course, we got the blame most of the time. Because most of the time it was the mom. And dad never did anything wrong. Dad didn't have anything wrong. It was always the mom, all right? But now we know that a lot of these genetic abnormalities and disorders, there's an organic base to it, all right? And why is that important? Because maybe later on we can find a way to cure them. Maybe find a way to cure them. Screen the egg. Screen the baby. And if they have this, see if we can fix it, all right? This would be hard to fix, but some other ones might be easier, all right? So, of course, because of this translocation, you get what we call an altered gene, right? That causes cancer. All right. Now, this is just recaps. I'm going to go through it really fast. Again, diploid cell is a cell that has how many chromosomes? 2. A pair of chromosomes, right? A diploid cell is that has a pair of chromosomes. Think of all the other cells, and it's a combination of an egg and a sperm, which happens to be haploid, right? They happen to be haploid. So this is a haploid cell. And notice when the egg is formed, 3 of them disintegrate, you only have one giant egg. And of course, genes are units of information about a specific trait. And their location on a chromosome is called that's your first test question. The location of a chromosome, location of a gene on a chromosome is called locus, right? The location of a gene on a chromosome is called I'm the one with concussion. What's your excuse, people? My head is blowing up. And of course, alleles are different molecular forms of a given. Remember, allele could either be dominant or it could be. And who always wins out?
Dominant.
Who wins out?
Dominant.
Right? In a straightforward Mendelian sense. And of course, the location of an allele on a chromosome is also called because alleles are genes. Don't confuse the two. It's not like there's two different things. You have genes for hair color, but they come in different alleles. You have genes for hair color, they come for different, all right? And of course, homologous chromosomes are identical in length and size, and they pair up during crossover, during prophase, right? It's just FYI stuff. Remember, these are homologous and they exchange pieces. These two can exchange, these two can exchange. Is that a bad thing or a good thing? It's actually a good thing. Now, if this one exchanges pieces with this one, that's an example of a bad exchange, all right? We'll talk about that later, okay? That's why genes are there. And of course, this is what the genome looks like. These are your genes and everything has been mapped. Now, if you're a female, you are XX, and if you're a male, you are a,
X1.
don't forget that, right? Normal female is a
X1.
X, and a normal male is a,
X1.
that's why a fetus that is a male is highly susceptible compared to a female fetus. And the reason is, this does everything. So if there is a defect in here, do we have a backup as guys? We have no backup. But in the case of women, you have backup. The ladies have a backup. So if something defective here, but this gene might not be defective. See how that works? Okay. Now, if they're both defective, homozygous recessive or homozygous dominant in some cases, then that's a bad thing. But the chances are much less than they are here. Sorry, guys. We suffer. We suffer. By the way, this chromosome right here, this chromosome, this is a human chromosome, number 2. It's actually 2 chimp chromosomes attached to each other. And I'll show you that when we do the human evolution. Because every chromosome, at the end, you have what we call telomeres. And in the middle region where the spindles attach is called a centromere. Well, this one has 4 telomeres and 2 centromeres. You have 1 telomere here, another telomere here. Then you have a telomere here and... No, telomere here. I'll show you the pictures later. And that's because 2 chromosomes have come together and they attached. And that's what separates the human lineage from the ape lineage. All right, that's why they have 48 pairs and we have 46. But we'll talk about that later in the semester. Now, sex chromosomes were discovered in 1800s, and mammals were brutalized again. X female, XY.
Male.
That's why these nucleoplaces that have X's on them, right? Right, triple X. Because guys have X and B. And of course, since we're guys, they have to repeat it 3 times for us to get it. So X, X, X. Oh, okay. All right. It's supposed to be a joke, people. We're supposed to laugh. Here am I. I have a concussion and I'm lecturing. My head is blowing up. Every time I walk, it feels like there's a bowling ball in my head. It's like moving from left to right, or right to left, and vice versa. All right. Now, in other groups, XX is actually a male and XY is a female. All right. Some other species have it. Some lizards. And there are some species don't have male at all. There are lizards that are all completely female. Completely female lizards.
How do they reproduce?
They reproduce by, depending on the ovulation cycle, either one behaves as a male and the other one behaves as a female. And they'll mount each other as if they're male copulating. And that just by acting on mounting, the female reproduce by parthenogenesis. And since you're only passing on X's, you always get female. All right? Now, when it comes to reproduction, let me go to my board over there in this crackhead room. When our chances... These are some easy questions. So if you happen to be a female, you are XX, right? If you have a male, you are
X1.
So if you were to do the Punnett square, notice the female is always donating X, right? The female always donating X, while the male can donate an X or a...
Y.
So if this sperm and this egg combine, these are what you get. Male or female?
Female.
Male or female?
Female.
Male or female?
Male. Male.
So it's 50-50, right? Chances of you having a male, a baby boy or a baby girl is what? 50-50. It is completely random. It's completely random. And who determines the gender of the baby? The mom or the dad? Dad. Moms have no say in what the baby's gender is going to be. It's the dad that donates it. Now, throughout history, who got the blame if they did not produce heir offsprings to the throne? The mom did, and they lost their head, right? Henry the 18th, King Henry in England, was it 8? Nasty guy, all right? He married, first wife, I think either gave him a daughter, never a son, or no kids at all. So off with her head. And then another one, same story, and he went to the Catholic Church and says, look, I want to divorce my wife. I want a son. And they're like, well, tough cookies, you can't divorce. And he said, well, I'll show you the Catholic Church. And he went and founded the Church of England. The Church of England started because Henry VIII wanted to have a boy. That's the whole foundation of that church. Kind of sad, but...
He didn't get it.
All right. And eventually he did get a boy. He did get a boy.
He got Queen Elizabeth.
Yeah, but he had a boy that got...
Oh.
His boy died of disease. I think Queen Elizabeth, the baby mother. All right? So, who got the blame? The mom. So King Henry sat through my class. None of this would have happened. All those women would have been safe. And by the way, there's nothing you can do. There is nothing you can do to control the gender of the baby. All right? Those of you going to be parents one day, the only thing you can hope for is the baby is healthy enough, right? 10 toes, 10 fingers, nose, ears, and they're healthy. There's no horns sticking out of them. They're not speaking in a demonic language like my child. But there's nothing you can do. If you go to Barnes & Noble, there are books that they sell people and they swear by it to control the gender. It's bunkers, all right? It's bunkers. What?
The Chinese schedule?
Yeah, the Chinese schedule, doing cartwheels after sex, all this other stuff. The only indicator is how fast conception took place. If the conception is early in the ovulation cycle, chances are it's a higher chance. Not 100%, there's a boy. And the reason is the sperms that carry the Y chromosomes don't live too long. So later on in the cycle, if the woman is getting pregnant, that means those Y chromosomes are already dead. They can't live on their own. The X chromosomes persist longer. So if the conception is later on in the cycle, then the chances are it's a girl. If it's early in the cycle, the chances are it is a boy. But that all depends how many Y chromosomes are produced, how many survive, etc., etc. All right? So just be happy. For now, don't have kids. That's my recommendation. Don't get married. How many of you single? Lucky people. Don't get married. But if you do, you fall and knock your head and you decide to get married, don't have kids yet. All right? Don't have kids yet. A lot of work. All right, so we talked about it. X and Y chromosome. That's what they look like. Look at that Y chromosome. Doesn't do anything. You know what Y chromosome gives you besides determining the gender? It gives you that hair that grows in your ears. You see these old men when they get old and have a lot of hair in their ears? Like really, like fluttering in the air when they're riding their motorcycle? That's a gene on the Y chromosome that becomes active once you hit the age of 50. There's a gene on the Y chromosome. All right? Now, to prepare an image of your chromosomes, this is going to be on your test, there's a technique called karyotyping. What you do in karyotyping is you stop the cycle, usually at mitosis, you add a chemical to it, and you stop the cycle. You take a picture of it. In the old days, actually, you take a picture and you cut it by hand. Now it's computerized. And then you just line up the chromosomes. You pair them up. So on the exam, if I ask you, what is karyotyping? Your response should be, karyotyping is a technique used to visualize chromosomes. Karyotyping is a technique used to visualize what? Chromosomes. All right? So let me show you how it's done, then I'll give you a short break. And I need it more than you guys. So look at, you take your cell, you add a chemical to it, some kind of an acid, it stops the cell from dividing. It arrests the development. It stops it. Then you photograph it using a microscope. Now, in the old days, you actually had to hand cut the picture. Now it's computerized. And then you arrange them in a diagram. And that's what you get. Chromosome number 1 all the way to 22. If you happen to be female, XX. If you happen to be a male, X. Once again, look at that Y chromosome.
So each chromosome looks different? They don't look the same?
Well, just because of the picture. But that, they carry the same genes. These two carry same alleles. Right? Now, you could have two dominants here, or dominant recessive, or recessive recessive. All right? This is another example of karyotyping. Is it male or female?
No.
Male, right? Because you have... By the way, I'm thinking of a business. It's not that hard to make karyotyping diagrams for people. Take a small drop of blood, you prepare it, you take the picture and you print it out and put it in a glossy thing and you frame it and you put it in your room. Wouldn't that be awesome? How much did you pay for it? I need to do a customer survey. That's a good business idea. And again, sex determination, it is 100%. Yeah, but who determines it?
The male.
Them. But it is 50-50. All right, it is 50-50. Doesn't matter. Chinese calendar, Mesopotamian calendar, the Mayan calendar, which is going to end pretty soon. You're always going to get what you get. All right, just be happy with the baby. All right. Now, hopefully your baby will be healthy and not ugly. There's nothing worse than an ugly baby. All right. Okay, go ahead and take a 4-minute break. Oh yeah, you heard me right. 4 minutes. I feel generous. There's no limit to my generosity. Sign-up sheet, how far has it gone? Did they unlock that door outside?
No, there's an exit now.
I think the school people didn't even know the school.
You got that right.
There's like, those are
Damn.
absences, correct?
Yes.
They were like back to back, and I think there might be a mistake, because I don't even think
Well. All right, let's get started. Sorry, now I hit her. So let's talk about the Y chromosomes, all right? Besides, what does Y chromosome do? Fuzzy ears,
I'm out.
using the remote control. Once, guys, even I catch myself. I'm watching the show I love, and I'm still holding the remote in my hand. Why the hell, It's like, all right. And it's not like, okay, even HBO, you don't have to fast forward through commercials. So, by the way, Walking Dead, so sad. And I have some questions after class. You guys have to answer them because I'm confused. Now, the most important thing the Y chromosome does, the most important thing it does is determine the gender of the baby. There is a region. It's called the SRY gene. SRY gene. If SRY is present, SRY stands for sex determining region of Y. If SRY is present, you get the formation of testes. Congrats, you have a baby boy. If SRY is absent, you get the formation of ovaries. Congrats, you have a baby girl. Now, this SRY gene doesn't kick in till later during the birth, right? There's no way you can tell if you have a boy or girl unless you go in there and take blood samples out and do genetic karyotyping, which is something no one recommends. Why? Because it's an invasive thing, right? And it will cause some serious problems. So up to 7th week, all you have is gonads. You don't know if you have a boy or a girl, externally or internally, all right? And even about 10th week, you can't really tell, but you can start seeing the formation of the testes and ovaries. And so what happens is, if it's a boy, literally the testes tube will turn, and you end up with the formation of testes. In the case of the females, the ovaries will literally turn like this, and you get the formation of ovaries. And all of that is determined by the SRY gene. Now, sometimes this process starts and something goes wrong with the SRY gene, shuts off. Something is wrong with that gene. There's a mutation on it. The default mode, remember you buy a computer, there's always the factory default mode, right? So it's like computers, you can always go to default mode. Well, the default mode in nature is female. The default mode in nature is female, because that's the stronger genetic gender. The female is the stronger genetic gender. And no, I'm not just saying that because I watched Oprah and she brainwashed me. Right? There's subliminal messages in Oprah shows, right? Never seen the show, but that's what I'm assuming. But that's the biology of it. And when that happens, so you start forming the penis and the testes and everything else, and then you go into a default mode, and then you go into a female mode, and that's the only time you get a true hermaphrodite. A true hermaphrodite is, and it's really rare. It is really super duper rare. All right? Some people, 1 in 600 maybe? Well, maybe it's not that rare. Now, for the longest time, what they did, right after birth, the doctor told the parents, well, congrats, you have a boy and a girl. And the parents had to decide, right there on the spot, if they wanted a boy or a girl. And they'll operate and they'll determine the gender and they'll raise the baby as that gender. The problem was, sex and gender are two different things. So a lot of these kids who were raised as a boy, they felt more like feminine, female. And opposite, you had girls that were raised as boys, but they, so it causes a lot of problems. A lot of them ended up with suicide. Now, they won't do anything. Now, they'll just wait till the child is a proper age and look at what the gender the child is affiliating with most, and then they'll operate, instead of the parents get to select. Because most often they went with the boy. There is a boy bias. Let's be honest here, all right? There's a boy bias. I had a friend who was doing cartwheels because he found out he didn't have a boy. Like, this is stupid. As long as the baby's healthy, right? I don't care if I have a boy or a girl. As long as they're healthy, that's what it counts. All right? And they're not ugly, too. That's important. So I do have my bias. But those are the rare cases, okay? Most of the hermaphrodites that we see is actually a pseudo-hermaphrodite. Most pseudo-hermaphrodites are technically female, but overproduction of male hormones causes enlargement of the clitoris and everything else, so it kind of looks like a male. But those are pseudo. True hermaphrodites on a genetic basis, and it's rare. It is rare, all right? And I think we're doing the right thing by not selecting the gender at birth, because a lot of these kids ended up committing suicide. Right? It's hard enough. But the X chromosome is it. It does everything. 2,300 genes. Most genes will deal with the non-sexual traits, but it's extremely, extremely important. So if there's something going wrong with this X chromosome, and of course the other X chromosomes, then there is a serious issue. And we'll talk about those today. Now, for the longest time, people just thought, well, X chromosome, okay, male and female, that's it. But no one could associate the X chromosome with a specific trait. And that's what we call linkage. Right? The term linkage means when you're associating the X chromosome with a specific trait. Remember we did those Punnett squares right there? Well, the XYs, then you put the trait on top of it, so you're carrying two things. And I'll show you how it's done. Now, one of the first studies was conducted by Morgan, and he took males that were homozygous dominant females that had red eyes, right? So female, red eyes, and crossed them with recessive males, and the males had white eyes. Now, how did he create this lineage? They just took fruit flies and then just bombarded them with radiation. And then created a mutant type, and then they just started cross-fertilizing the mutants together, and they created a true breeding lineage for the white eye color. Now, this is what happens with this guy. Let me show you in the Punnett square, because you'll have questions like this. He crossed a male with a white eye with a female that was red eye, homozygous dominant, and all the females were heterozygous and had red eyes, and all the males were red eyes. So let me show this right here. This is how you do it. Female, homozygous dominant for what eye color? Red. So you put the X chromosomes and then you put the R's on top. See how that works? Mm-hmm. And then the male is white eye, so he has a recessive allele, right? You put the lowercase r. Do I put anything on the X? No, because this is an X-linked trait. See how that works? So when you're doing the fertilization, put the female here, put the male here. These two cross, right? These two cross. These two cross. Now, male or female?
Female.
What eye color?
Red.
Male or female?
Male.
What eye color?
Red.
Male or female?
Female.
What eye color?
Red.
Male or female?
Male.
And that's what those results are. That says heterozygous or interchangeable. Just male. There's no heterozygous because you only have. So this is how these Punnett squares work, okay? So when I'm talking about an X-linked trait, and I'll tell you, you have to put the trait on the chromosome, all right? So on the exam, if I ask you what percentage of the daughters are colorblind, red eyes, your response should be? What percentage of daughters are red eyes? 100%. What percentage of the boys are red eyes? What percentage of the offspring are red eyes? 100%, right? So be careful if I'm asking for a boy or a girl. Now in this case, all the numbers are the same, right? But there are cases that are not. We'll talk about that. Okay? Any questions so far? So all F1 offsprings had red eyes. And then he started crossing those guys. He started crossing them. And he did this variation too. He took a female that was homozygous recessive. In other words, the female had two lower cases R's and crossed it with a dominant male that had red eyes, right? In that case, all of the offsprings that were male had white eyes and all the females had red eyes. So have had red eyes and have had white eyes. So what does that tell them? So what does that tell them? Go ahead. Yeah. The color of the eye is determined by the... Because if the mom is recessive, right? So she is passing an X with a recessive allele to the boys, right? Because the boys always get the X from the mom, right? The boys always get the X from the mom. And that's why the boys are always, in this case, that's what we call boys, the male flies are always colorblind. The mom could be the carrier. Can dad pass on the colorblindness to his daughter? Can he pass it on to his son? Right? Because dad is passing the Y to the son, right? And he might pass the colorblind X, but if the mom is normal, then it washes out. That's why moms are always carriers when it comes to X-linked traits. The female is a carrier when it comes to X-linked traits. So Morgan crosses show that relationship between the sex and the eye color. Female can have white eye color only and only if they are homozygous recessive. Only if they are homozygous recessive. So he concluded that the gene must be on the X chromosome. This was the first time that they established a relationship between an X chromosome and a specific eye color. Okay? I'm going to do it on the board now so you guys can see it, because I want to make sure you get this. It will be on your test. Not this scenario, but something similar to it. So, look at, as you young people like to say, the mom is colorblind, right? So you put two X's and dominant R or lowercase R? Uppercase R. Lowercase R, right? Because she has white eye color, right? And you cross her with a male that has normal vision, right? So if you do the Punnett square, and then I'll put these here, combine, so notice, this daughter, because of this dominant allele here, will have what eye color? Red, right? What about this one? Red. What about these boys?
White.
They're both going to be, because the boys always get the X from the mom. And if the mom is colorblind, that means she has the recessive allele, both of them. Right? That's why only the mom can pass on an X-linked trait. Colorblindness is only passed on by the mom. Hemophilia, only passed on by the mom. The dad is passing on the... So on the exam, if you do something like this and they ask you what percentage of the offspring are colorblind, you look at the whole picture, right? And white eye color, you get the three. That means 50%, only half of it. What percentage of the boys are colorblind? You say white eye. It's the same thing, right? What percentage of daughters have red eyes? You say 100%. Right. So make sure you go practice these X-linked traits. It's similar to the other one. Only thing you have to do is track the X's and Y's. You have to look at the gender too. You have to look at the gender. God, I wish I could cancel class. Don't worry about full and complete linkage. Now, remember we talked about crossover during prophase, homologous chromosome exchanging pieces? Sometimes you get traits that are linked together. So you have this gene A, gene B, gene C, and gene D. And these two traits are linked together. Wrinkle seed and a tall plant, for example, versus red flower and thorny bushes. Whatever. Want to get the general idea? Now, if the crossover takes place, the chances of disrupting this link between these two is much higher than disrupting it between these two. Okay? So do you see the distance playing a role here? If the genes are closer to each other, on the chromosome, the chances of them being crossed over together is much higher than they are far apart. If the genes are far apart, the chances of you breaking this linkage is much higher than it is here. Alright, so keep that in mind. So the distance also plays a role when it comes to linking of different traits. Now, up to this point we talked about fruit flies, Pi sensitivum, garden pea plant that Bendel worked with. They use Cinerotitis elegans. These are nematodes. You can even use bacteria because they can produce quite a bit of it. But how can we do this with humans? Which woman in her right mind will agree to have 50 babies in order for us to properly establish a relationship? Any ladies here willing to have 50 babies? I don't think so. Unless you're kidding, you raise your hand. So that's why what we do is pedigree charts. Pedigree charts is using symbols and looking at genealogy and tracking people. All right? Some people can track their genealogy back to 1600s, 1500s. Those are the royalty. Anyone here can track their family to 1800s? Okay. I had students in the past that they can track from like 17-something. They have the family charts, or great-great-grandfather. They have a chart. If that's the case, then you can actually keep track of it. Now, the best way to establish pedigree charts, I forgot what her name was. She was a grad student. And she went to this, it's not an Amish. They're not Amish. They're not, I think they were Hurrites. One of those tribes. Remember that TV show they had, Meet the Herd? Because they keep good pedigree charts. You know, who's who and what are the traits. And they marry within themselves. So she used a pedigree chart to track a specific trait, all right? Now, with pedigree charts, you need to understand the symbols. This is important. If it's a circle, it's a female. So circle is female. If it's a square, it's a male. And if it's completely full, dark, that's an example of a person that is showing the trait. And that means they are affected. And if it's partially full, that's a person that happens to be carrier. All right, so let me show you the pedigree chart. This is a female, circle, male is a square. If it's completely full, that means the trait is present. If it's halfway full, that means the person happens to be a carrier. But sometimes for the carrier, they also use a star. Sometimes for the carrier, they also use a star. Now, this is a pedigree chart that she has established for just one family, and she did it for everybody. Now, she's tracking what we call polydactyly. Polydactyly is having 6 fingers, or 7 fingers, or 6 toes, or 7 toes, all right? It doesn't have any health issues with it, but, you know, makes this little piggy kind of thing confusing, because you run out of piggies, right? Or you have extra piggies, and you run out of orders. But she tracks it, and it's very common in groups that have a lot of inbreeding. If it's a closed society, and people are just constantly marrying each other, cousins and cousins and cousins and cousins and cousins, the chances of passing on this trait is much higher. So, she tracked them, mom and dad. Were they normal? Yes. They had a daughter, and that daughter had 5 fingers and 6 toes. That's what it means. 5 fingers and... Now, that daughter married a male, and they had 1, 2, 3, 4, and 5 kids, right? How many of the kids were daughters? 1, 2, 3 daughters and 2 boys, right? All of the daughters show the trait. Do you see that? While the boys, one was normal and the other one was carrier. Now, this carrier got married, and then they produced offspring, but notice it's a triangle. That means they don't know if it's a boy or a girl. They probably left the tribe or whatever. So, and they look at the name and says Dana. Well, you don't know Dana is a male or female. So they kind of know, and then they suspected one of them was probably showing the trait. But notice, the trait shows itself more often in the these daughters. This one was just like her mom, 5 fingers and 6 toes. But these 2 daughters had 6 fingers and 5 toes. Okay? And they got married, and then you got a bunch of kids. Holy cow! Look at this. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And this one had only 2. This one had one daughter that had 5 fingers on one hand, 6 fingers on the other, 6 toes on one leg, and 7 toes on the other. Right? She got married to a guy, and then they produced 2 boys that had the trait. These 2, they lost their tribe. Probably baby died. I think that's 12 or 6 weeks or something like that. And I notice most of the boys got it here. So what is this showing you? That this trait is being passed on mostly by which chromosome? X or Y? X. Mostly passing on by the X. It's the mom that is passing it along. Now, do you expect it to be a recessive trait or a dominant trait?
Dominant.
You sure?
Recessive.
Probably, because if it's dominant, all of these kids get an X, don't they? See how that works? And they get the X, that means all of them should show it. So keep that in mind. If I give you a pedigree chart on an exam, and I will, you have to be able to interpret it and tell me if it's a recessive or dominant. If it's dominant, that means it's always showing itself. If it's recessive, that means it only shows itself sometimes. When it's homozygous, or in the case of the boys, it's one time, right? One shot. Which brings me to the term called genetic abnormality. Something like polydactyly, all right? You know, change different numbers of toes or fingers. They don't cause any, generally speaking, no serious health issue. If you have 7 toes, is that a health issue? No, it might make buying shoes a bit difficult, right? Or you have an extra finger, or maybe it make you a better catcher or goalkeeper, I don't know, right? Makes flipping people off different now. Extra. Double flip off, all right? But health-wise, is there any issues with them? No, right? And that's the term we use for genetic abnormalities. Genetic abnormalities don't have any health issues with them, but you're expressing the trait slightly differently than, quote-unquote, what normal is, all right? And by the way, this trait is subjective. Some cultures worship having extra fingers, all right? So it's completely subjective. It's completely subjective. versus genetic disorder. If you hear the term genetic disorder is bad. Genetic disorders have serious health issues associated with them. Again, genetic disorders have serious health issues associated with them. Yes?
Wait, the last one was genetic
Abnormality.
abnormality.
Okay? Genetic disorders have serious health issues with them, and we're going to talk about all those disorders shortly. And you need to know them on the exam, okay? So, abnormalities, not necessarily bad. Disorders, it is bad. Baby is born with a tail. Is that a bad thing or a good thing? It's subjective, right? Is there any health issues associated with it? It makes it easier to carry. Grab it from the tail, all right? But in reality, there is no health issues associated with them. We'll talk about leukemia and all that stuff later, okay? So keep that in mind. Now, why are these guys not disappearing? Well, sometimes you get mutations. You're smoking, you're next to a radioactive facility, you drink alcohol. All of this stuff contributes to the mutations in your genes, and you might create a mutant that is bad, and then you pass it on to your offspring. All right? And of course, if you happen to be heterozygous, the harmful one, if it's recessive, is masked by the dominant, right? So you might not have the trait, but you can pass this on to your offspring. Okay? Now, when it comes to these genetic disorders, some of them are called autosomal recessive. Autosomal recessive. That means which allele is bad? The dominant allele or recessive allele? Recessive allele. Alright? So if you have two parents that are carriers, so this is normal, this is bad. Normal and bad, right? And if they cross-fertilize, notice they will have a 1 in 4 chance to have a normal baby. If this normal sperm mixes with a normal egg, right? There is 50% chance that will produce babies that are normal, but they happen to be what? Carriers. And there is 25% chance to have a baby that happens to be affected. Yes?
So when they're a carrier, that just means that when they end up reproducing...
And if two carriers reproduce, then it's going to be 1 in 4. Now, if a carrier marries somebody that is completely normal, then you produce other carriers, but you never produce the trait. Right? Now, an example of autosomal recessive trait, if I ask you an example, is galactosemia. Very common in Northern European descent. All right? Now, we knew that was not a child with my son because my wife is Northern European, but I'm Middle Eastern descent. We don't have this. So the chances of me being a carrier was pretty much zilch. So I calculated this one out of the equation really fast. This is a disorder that you don't have the proper enzymes to break down lactose. This is not lactose intolerance, people. Lactose intolerance does not kill you, does it? No, it kills the people around you. Lactosemia, true. What it causes, it can cause blindness and it can kill you. You don't have the proper genes to break it down. You end up building up lactose-1 phosphate in your system, which is pretty nasty. You don't have the proper enzymes to break it down. All right? So this is an example of an autosomal recessive. But you have other ones that are called autosomal dominant. You have other ones that are called autosomal dominant. Now, which allele is bad here now? And which one do you think is nastier? Autosomal recessive traits or autosomal dominant? Dominant. Because if you have a dominant allele, that's it, right? If you have a dominant allele, that's it. So, usually an individual that is homozygous dominant, they don't even survive. Homozygous dominant, chances are they die. So usually you either normal, homozygous recessive, or you happen to be a carrier. And you pass it on, and of course half of your offspring could be affected. Half of your offspring could be affected. Now my question to you is, this is an autosomal dominant. Why are these individuals, or how are they surviving, thus being able to pass on their genes? What is that telling you? When is the trait showing itself? Early in life or later?
Later.
The trait shows itself later in life. That's why most people reproduce, and after that they found out, oh, I have this trait. All right? Such as Huntington disorder. All right? Neurological disorder. Most people who have Huntington disorder, you won't know till after the age of 30. And most people have reproduced by the age of 30. They have passed on the gene to their offspring. See how that works? So with these autosomal dominant traits, they don't show themselves till later. They don't show themselves till later. Okay. That's why 30 is a milestone. Once you pass 30 up to 35, if you're not showing any of these neurologists, you pass quite a few of these disorders. And then there's another one up to 40, all right? So that's why it's worthy to celebrate. Not 21st birthday. It's 30, 21, all right? So people often pass on the allele before they know. It causes an involuntary movement of the nervous system, deterioration of the nervous system. You become immobilized, you can't do anything, and eventually you die. Very similar, but that's not autosomal dominant. ALS, right? What is ALS? Lou Gehrig disease, right? Now, Lou Gehrig is pretty nasty because you just lose muscles. You just lose muscle. A good friend of ours passed away last year. But the brain is there. The brain stays exactly the same. That's why it's probably the cruelest way to die, if you ask me. Because you just lose muscle. You can't move anything. You can't speak. You can't breathe on your own eventually. But your brain is the same thing. And this individual actually was in the genius rank. He was a top 1% in the genius. I used to joke with him, like if I get into Who Wants to Be a Millionaire Lifeline, because he knew everything, all right? He was truly a genius among genius. But imagine how cruel that is. Your brain is still there. You just know you can't do anything about it. That's why I show symptoms. I'm going my own turn, all right? It was pretty bad. I know it's selfish, but that's how it was. Achondroplasia. This is another form of autosomal dominant allele. If you're homozygous dominant, stillbirth, baby don't survive. But if you're heterozygous, you get a form of dwarfism. You have short arms and legs relative to the other part. Your torso and your head is quote-unquote normal, but your arms and the legs happen to be shorter. Remember that TLC show, Little People, Big World? The mom, right? Head and torso is normal, but the limbs are short. That's called achondroplasia, and it's a form of autosomal dominance. All right? You pass it on. And then, of course, you have what we call X-linked recessive inheritance. These are the ones that are passed on by only the mom, and they happen to be recessive. They're passed on by the mom, and they happen to be recessive. All right? An example of that, colorblindness. All right? And there's a distinction between colors. I had a teacher that was colorblind, so his wife had to sew like dots in his socks so you can match them up. 2 dots is like brown or whatever. All right? I think it was red, green. I don't know what colorblind he was, but there's different shades of colorblind, and we'll test for that today or this week in the lab. Okay? Hemophilia, blood clotting disorder. You bleed to death. All right? 1 in 7,000 males have hemophilia. It used to be very common in the European Royal Society. Because the European Royal Society, they're all related to each other, right? Pretty much they're all grandchildren of Queen Elizabeth. All right? They were all related. So they, all cousins begat cousins kind of thing. And that's why the incidence was really, really high in the royal family. Not as much anymore, because they marry outside. But if you're constantly marrying within your family, the chances of passing on that trait is even higher. And we will work on colorblindness in the lab. Learn that. It's important, because you will see it in your lecture exam too. Okay? But this is the same thing, but now we just have to put the colorblind allele, like a lowercase d here. We'll talk about that later. And there's a condition called Fragile X syndrome. By the way, all these disorders, make sure you know them, what they are. You'll see them on your exam, 80% of them. Fragile X, it's like a recessive disorder. And you have this mutation. Look at this X chromosome right here. It causes it to look like it's flimsy. That's why it's called the Fragile X syndrome. And the disorder causes you not to produce the proper proteins for the normal brain development. So you end up with some type of, it's not really Down syndrome-like symptom, but it causes some kind of a mental retardation. There's different levels to it, but it has to do with that X chromosome right here. There's a mutation right here. Hutchinson-Gifford progeria, we don't really know what causes it. Could be a spontaneous mutation. Some people say it might be dominant, but it causes aging to occur really fast, all right? These 2 kids are like 9 years old, but they look like they're 70. They're 9 years old, but they look like 70, because the body, the cells just age really, really fast. This is not Benjamin Button. I've never seen that movie. I have no inclination of seeing that movie. Not World Z, yes, for sure. But the baby is born, and they just age really fast. They age really fast, and they tend to die in their early teens, all right? They tend to die in their early teens. This young man right here was actually an actor. His last movie was with Marlon Brando, what's his name? Island of Dr. Moran, what it's called? It's like a horror movie, but the remake of it. He was one of the characters in there. Now, a lot of people are studying it, because if you can stop this process, what can you slow it down? Aging, right? And how much money we spend on anti-aging products. I mean, just think about it. How much money we spend? From makeups to Botox to firmers and collagen injections or facelifts, or name it, right? So if somebody can figure out how to stop it at a genetic level, they will be super duper rich, right? Problem is we don't know how to start, so it's very difficult. But some people are studying it. Now, up to this point, I talked about specific disorders. Now, I'm going to talk about things that can happen on the chromosome. All of this will be in the text, so it's important. Pay attention, all right? These are the things that can happen to the chromosome itself, all right? Mutations, we call these mutations. One of them is called duplication. Duplication. When you think of the term duplication, what does that mean? To make doubles, right? Well, in duplications, part of your chromosome will just duplicate itself. Maybe twice, maybe 100 times, maybe up to 1,000 times. So let's say you have a gene, adenine, thymine, hypothetically, right? That adenine, thymine is repeated 1,000 times because that genome is duplicated. So imagine you have these genes, gene D and E. You can duplicate twice, it can duplicate 3 times, it can duplicate 100 times, right? Now, duplication is not necessarily bad because the original one is still there, right? And if the original one's still there, that means the function is not lost. But guess what? Now you have backups that mutations can accumulate in and maybe cause a forming of something that is useful, right? A lot of the complexity we see in the snake venom is the result of duplications. Snake venoms have duplicated. Part of the gene that codes for that venom has duplicated itself. And then these other parts, they can build up mutations in them because you're not influencing the functions, and eventually you might get a new trait, in this case, more potent venom. Right, more potent venom. Mojave green, their venom is actually getting nastier. Right, so don't get bitten, people. Another mutation is called inversion. Part of the chromosome breaks off, twists itself. So imagine you take a sentence, cut it in half, and reverse the words. Is that going to get confusing? Right? Just reverse all the letters. Wouldn't that make it confusing? Mm-hmm. And this is a nasty mutation that can occur. What can cause you to break your DNA? Well, go dance in front of an X-ray machine. Okay? That can cause it to break down. Okay? Go dance in front of an X-ray machine. Translocations. When one gene from one chromosome picks up and it moves and lands in another chromosome. Remember we talked about that Philadelphia chromosome we started with, right? That's an example of translocation. You're relocating one part of the chromosome to another one. Now, if this occurs between homologous chromosomes, so be it. That's fine. But if it's between non-homologous, then that is pretty bad, right? Because they're not similar. So imagine these two chromosomes exchanging pieces. This part goes here, this part goes here. Now you have what we call an altered gene sequence. It causes serious problems. Deletions. You can lose a whole part of your chromosome. Go dance in front of an X-ray machine, the chromosome gets snipped, and you just lose that part. Losing part of your chromosome, you might lose important genes, right? And if you lose important genes, what's going to happen? You're screwed. You're screwed. If that gene is important for your...
How do you say that could happen, though?
Chemicals can cause it, radiation can cause it, and sometimes just chance. Just chance. There are enzymes that can break it. Okay? But of course, the more exposure to environmental toxins increases our odds. Okay? Now, the term aneuploidy, a normal individual is 2N, right? 2N. In aneuploidy, you're 2N plus 1. That means you have an extra chromosome, or you have 2N minus 1. That means you have lost a chromosome. This can cause Turner syndrome. This can, operative word, cause Down syndrome. Now, how do you get that? By the way, most of these are miscarriages in humans. but sometimes they go through. Well, what happens is, remember we talked about prophase and metaphase 1, homologous chromosomes pair up and then they separate, right? You expect 2 to go here, 2 to go here, right? But you get what we call non-disjunct separation. 3 go up there and only 1 goes down in this scenario. And of course, when you create the haploid eggs, now you have eggs minus a chromosome, right? And you have eggs with an extra chromosome. And if these eggs happen to survive, the body doesn't correct itself, and if this gets fertilized, you end up with
Down syndrome.
Down syndrome. Gets fertilized, you might end up with Klinefelter syndrome, or this might end up with Turner syndrome, all right? That's what happens. Why non-disjunct? Well, mistakes do happen. When the spindles separate, there's no law to it, just mistakes happen. And the older you get, the chances of non-disjunct separation is much higher. Once you hit age of 35, the odds of this happening is much higher than if you are 19 or 20. That's why if you're older in conceiving, they actually have to go through more screening process, because as older you get, actually any pregnancy over 35, my wife was 35 when she got pregnant, actually she just turned 35, all right? It's considered geriatric pregnancy. Now what is the term geriatric pregnancy? Old person. Anything over 35 is actually considered geriatric pregnancy, so you have to go through multiple screenings. And of course, more women are waiting later to have kids because, you know, you're professionals, you're going to school, there's a lot of work. So a lot of professionals know what they do. They store their eggs. They're viable eggs when they're 20, 24, 25, and they want to conceive. They go take their eggs out of the bank and then fertilize them and implant them, and they get, because they're worried about this happening. Right? But the older you get, the chances are higher. Yes?
Does that work for men as well? Like,
not really. Men, you always are producing sperms. There's abundance in numbers. And the Down syndrome, all that stuff is mostly mom, not dad. Now, they suspect if dad is a smoker, they might cause other issues, but not something like this. All right? Yeah, you produce so many sperms, it's like a dilution effect for millions of sperms. With female, just 1 egg. All right? By the way, polyploidy doesn't happen to humans. This is 3N, 4N. Very common in plants. And humans die. No human ever survived. The longest one was a newborn that survived for a couple of hours. It was a 3N. So that's like all of these going to one side. So you have a 2N egg to start with, and you get fertilized, now you have a 3N. Rare. Okay? So, Down syndrome, trisomy 21. Don't forget these two. By the way, Down syndrome, there's a scale to it too. Right? There are Down syndrome kids that can be functional in the society. They can hold jobs, they can do things, become actors. Remember there was a kid, Corky? There's a TV show in the old days. He had Down syndrome, and he was actually an actor. So he functioned. But of course there's extreme end too. There are Down syndromes that are severely handicapped, and they need 24/7 care. Yes?
Aren't some people with Down syndrome, like, they're more intelligent in other aspects? Or they have, like, a better focus or something?
Down syndromes, generally speaking, they're good at repetitive tasks. Putting beads together, making something repetitive. I think you're confusing that with what we call autism, which we really don't know what causes it. You know, autistic kids are super duper with math, but when it comes to social skills, you know, they're missing and so forth. And really don't know what causes autism. It is not vaccines, for sure. And all the studies point to that out. There was only one study that suggested that vaccines cause autism, and that paper was retracted because the data were false. Actually, they were fabricated. But, of course, Jenny McCarthy still says, don't vaccinate your kids, and people don't vaccinate, and that's the sad part. Yes? It is a form of Down syndrome, too. It's Asperger, too. They're a little bit more functional. They can do math. We have colleagues that the kids can do math stuff, for example, really good math, but ask them to write an essay or something different, they can't do it, all right? They can play poker better than anyone because they can keep track of the cards, but they can't go outside and shop on their own, all right? Okay, there's 2 minutes left. Just to remind me, we stopped at Down syndrome. There's not much left. Okay. I'll see you guys on Wednesday, assuming that I don't have any traumatic brain damage.
Are we having a clear
Well, no, let's go. Next week is Thanksgiving. That way people have to show up and not